Hello, we're glad you've joined us for this live webinar, Analysis of Surface Antigens on Exosomes using the Invitrogen Attune NXT Flow Cytometer. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Brought to you by Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science, whose mission is to enable customers to make the world healthier, cleaner, and safer. Thermo Fisher Scientific helps their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. For more information, please visit www.thermoscientific.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience any technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Steve McClellan, the Manager of Basic and Translational Research Operations at the University of South Alabama Mitchell Cancer Institute. He also serves as Chief of the Flow Cytometry Core Laboratory. Steve has more than 30 years of expertise in advanced flow cytometry and cell sorting. He has worked in basic and clinical research in the areas of cancer biology, stem cell therapy, transplant immunology, and xenotransplantation. For the past 12 years, his lab has been conducting research on cancer stem cells and circulating tumor cells. The aim is to develop better methods of purification, culture, and analysis at both genetic and functional levels, and using cancer stem cells in high-throughput screening drug discovery. Exosomes are the latest addition to the lab's research efforts. His complete bio is found on the LabRoots website. Steve McClellan will now begin his presentation.
Thank you. I would like to share with you some of our recent progress in using the Attune NXT to analyze surface markers on exosomes. Some of the first published reports on exosomes date back to the mid-'80s, and the field actually began in the world of platelet biology and reticulocyte maturation. Since that time, the number of references in publications talking about exosomes has grown exponentially. Around the turn of the millennium is when we see the first reports of using flow cytometry to actually look at exosomes. And in the last 5 to 10 years, we've seen yet another explosion in those publications due to the improvements in the sensitivity of our instruments and also highly defined protocols to add rigor to this, what I consider to be a rather challenging field. So let's talk about exosomes and microvesicles. These are actually vesicular particles that are shed from cells comprised of a lipid biolayer and containing representative cytosolic content from their cell of origin. This can be DNA, RNA, especially microRNAs, and proteins. Exosomes have been shown to affect intracellular communication. I think early on they were actually considered to be garbage bags. People thought that the exosomes were being used as a way to shed extracellular debris and waste products. But we now know without much doubt that this is how cells communicate with each other. We'll talk a little bit more about that later. The surface markers on these small exosomes can also be representative from the cell of origin, matching some of the same epitopes on the surface of the cell of origin. We generally consider exosomes to be the smallest of these particles, ranging in size from 30 to 150 nanometers. The next larger portion of this fraction of vesicles are what we call MVs, or just microvesicles, and those are going to range in size from about 150 to 1,000 nanometers. In the last 10 years, exosomes have been implicated in just a myriad of different disease processes. Almost every disease process has had some type of work looking at the exosomes related to it. We work on cancer, but especially in cancer, exosomes are very critical. We now know that cancer cells secrete upwards of 20 times more microparticles than do normal healthy cells. And in the setting of cancer, these actually have direct effects on the healthy target cells that they bind to once shed from the cancer cell, doing a myriad of things, promoting growth and metastasis, vascularization of the new growth of the tumor, and also, most importantly, contributing to immune suppression. We have evidence that the exosomes leave the tumor cells and actually interact with the T cells to try and dampen some of the immune response. So this little cartoon kind of depicts two different ways of how these are formed. So the larger microvesicles actually bud off of the surface, literally pinch off and just release away from the cell. Hence, they're much bigger. True exosomes are developed through an endocytic process. So small clathrin-coated pits actually invaginate from the exterior of the membrane, forming what we call an early endosome. That little packet matures into then what we call a multivesicular endosome. And at that point, those exosomes can either be released back to the surface if that MVE is actually directed to fuse back to the membrane and just revaginate, I guess you'd say, and expose the exosomes to the exterior surface, or lysosomes will bind onto that to destroy the contents of the MVE. So there are several ways to isolate exosomes. We'll start with precipitation. Well, let me start by saying that there are really two primary sources that you're going to use to isolate exosomes. For basic research, you can easily use culture supernatant. Cells grown in culture constantly shed exosomes. It can be a very nice clean source to look at specific markers or microRNAs of interest. There's a variety of body fluids that people have isolated exosomes from: urine, blood, which would be really plasma. And that distinction is important. Clotted blood, which would be referred to as serum, is not a good product to use. Many of the exosomes get trapped in the clotting process, and so your yield would be far lower if you used serum versus plasma. They've been found in saliva, cerebrospinal fluid. Just about any fluid in the body is going to have exosomes in it. So you can precipitate these. There are, in the last 5 years especially, we've seen an explosion of kits on the market that actually allow you to precipitate these small vesicles. In our hands, these kits don't provide very good purity. They're quick. Usually within an hour you can isolate the little particles. But we've seen that they might not even be suitable purity for even doing Western blots. We certainly have not had any success using precipitation kits for purifying them to do flow cytometric analysis. They seem to be very sticky after whatever chemicals are used to do that precipitation. So pretty much the only thing we found them useful for was really working towards DNA or RNA isolation at the end of that process. So size exclusion filtration can happen in two ways. You can use membranes with a known pore size, say 100 micron to do the filtering out the largest vesicles, and then the exosomes smaller than 100 microns would come down through, or nanometers, excuse me, would come through. We've played with this a little bit. They tend to clog pretty quick, and so I don't think it's a very time-efficient way of doing it. I think if you're really going to do size exclusion, the best way would be through chromatography. Small columns with some sort of chromatography material in it that really can just do size exclusion. So the smallest particles run through first, and you collect fractions. And those can provide very high purity. Sometimes you're worried that you might not get full recovery, so your yield might be a little lower in that method. And it could certainly never be considered quantitative, because each sample might not travel through the chromatography bed quite the same. But they do come out very clean in that respect. So we like that for flow cytometry. Back in the olden days, when people first started trying to do flow cytometry, immunoaffinity beads were really the way that everybody did this. So small magnetic or polystyrene beads coated with specific antibodies that bind exosomes. We'll talk a little bit more about these markers later, CD9, CD63, and CD81, that capture and localize the exosomes onto the beads. You can then wash and have a very pure preparation. You could go directly to isolating the DNA, RNA, or proteins from those exosomes at that step, or you could use more antibodies to label the surface of those exosomes that are now immobilized onto the bead and do flow cytometry. And that's really what they were aiming for before the flow cytometers became sensitive enough to actually resolve the individual particles, which we'll get to in just a second. But in the early days, you really could only see small beads on the flow cytometers, and that was really the only way you could qualitatively assess what was on the surface of an exosome, by using the bead capture method. We and many others consider differential centrifugation to be probably the best way to isolate exosomes, at least with very good purity. It's highly time-consuming. As you can see on the right, there is a set of successive spins. So each preparation will take a minimum of 6 hours, really, if you push right through it. You can do several, well, I can't do too many because most of the centrifuges only have 6 tubes in them, so you can only process 6 things at once. So it is time-consuming, but in our hands, this provides probably the most pure product that we can work with, especially when we're looking at doing flow cytometry where they're not left with any kind of sticky coating, and we don't get too much non-specific antibody binding. As we go through each faster spin, you generate the different levels. So first we're going to rule out the cell and cell debris particles at the first spin. The large vesicles from the second spin are considered apoptotic bodies, and another type of vesicle called a large oncosome. We're not going to talk too much about those today. The next 16,500 G spin generates the medium size, what we would consider microvesicles, in that 150 to 1,000 nanometer range. Then we take that supernate and put it into the ultracentrifuge for 2 hours, and those are the exosomes, the small vesicles that are actually in that range of 30 to 150 nanometers. Leftover juice is really just soluble material. There's not much left after the ultracentrifugation spin in the supernate. So early on, when we started working with exosomes and trying to do flow cytometry with them, we came up with an idea to use ultrafiltered solutions to reduce the burden of debris in those solutions that we use to prepare the exosomes, and also in the actual focusing fluid or sheath fluid that runs through the flow cytometer, which helps us to improve background noise. Traditionally, sheath fluid is only filtered down to about 100 nanometer particle size. And that's roughly the same size as exosomes. By using it's a PES membrane that has only 30 nanometer pores. And that way, all of the solutions really don't have any debris particulates that are larger than the size of the particle we're looking for. We believe, and we've noticed a huge significant difference in sensitivity of the instrument when we're able to do this ultrafiltration, and think that it's really a key point in making this much more successful. So once you've done whatever method you're going to do to isolate your microvesicles and exosomes, you need to be able to quantitate really what size particle you've isolated. You've got to make sure that what you're calling an exosome is truly the right size. There are several ways to do this. Unfortunately, all of them involve use of yet another instrument to judge size. Probably the most popular at this point in time is NTA, nanoparticle tracking analysis. These instruments actually use a laser to illuminate the particles in solution, but it's an imaging-based system. So it actually watches the vesicles move around in solution, and it's using the old-style technique of Brownian motion. So the laws of physics that cover Brownian motion are how it develops algorithms to actually calculate the size of the exosomes. It's very quick, and it can also at the same time give you a relative concentration of the number of particles per milliliter. So like I said, that's pretty much become one of the most popular systems to use for sizing exosomes these days. A newer version of a sizer is TPRS, Trace Resistive Pulse Sensing, that's a mouthful, is actually based on the old Coulter principle of sizing particles, just on a nanoscale. So in this method, a membrane has a very small nanometer hole in it, and every time a particle crosses through it, an electrical charge is developed from one side to the other side of the membrane, and that is responsive to the size of the particle. It's highly accurate. It also gives you a very accurate method of concentration. These instruments haven't been around that long, and I think that over time they're probably going to overtake NTA because, in my opinion, I think they're probably quite a bit more sensitive. DLS, dynamic light scattering, is also a laser-based system. The nanoparticles are in suspension, the laser hits, and as light diffracts off of and scatters off of the particles, there are a number of different detectors at different angles that are able to calculate the size based on the type and light scatter pattern. These are also considered to be pretty accurate, but the problem with DLS is that it does not give you any kind of idea of the concentration of the particles in the solution. And last but not least, the gold standard. If you want to measure the size of something, you look with an electron microscope. And that's true for exosomes as well. Traditionally, transmission electron microscopy, TEM, has been used. The newer modality of cryo-EM is probably better. It helps preserve the structure a little better, but either work fine. And really, many people want you to at least show that to make sure that you're correlating and that the other instruments that you've used actually are accurate. So we do, from time to time, submit samples for EM just to double check and make sure we're on track with everything. Quickly, I'll mention, I think I will say it later, but here at our center, we use DLS for sizing our exosomes. So let's talk briefly about the Attune NXT. This is kind of a different beast. The Attune NXT is the only system on the market that uses acoustic waves to actually focus the cells into a very tight and uniform line as they flow past the laser interrogation point. All other flow cytometers use the tried and true principle of hydrodynamic focusing, which uses high pressure sheath going through the flow cell, which causes a swirly effect kind of and forces those cells into a tight line. When you look at acoustic focusing versus hydrodynamic focusing, you can achieve much tighter peak CVs compared to traditional cytometers. It really is aligning those particles much more precisely as they cross through the laser beam. And another advantage of the hydrodynamic focusing is that it gives us very, very discrete control over the speed with which our particles are flowing past the laser beam. So the benefit of that is that the Attune can actually process samples much faster than just about any other cytometer on the market. For us, the flip side of that is that we can now slow them way down. We run at the very slowest dynamic rate, which is 12.5 microliters per minute of sample injection through the laser beam. And what that allows us to do is actually increase, maximize the dwell time that each little particle spends as it crosses that laser beam. So the slower you go, the longer, and of course we're talking microseconds, but it's enough to make a difference in the amount of photons that can be captured off. So it really does, running slower in this system really does increase the sensitivity in that you can collect more photons from each particle and see a brighter signal. So not only do we have the fluidics part, but the Attune does have fairly high-powered lasers. It can provide 50 milliwatts on the 405, 48, and 561 nanometer lines, and 100 milliwatts at the far red 647 line. And it does that in combination with something called flat top beam shaping optics, another mouthful. Really what that means is that instead of focusing the laser into a very tight spot, which the cells may or may not correctly intersect, the laser beam is actually 10 nanometer by 50 nanometer wide. So what it means is that the alignment of the instrument is almost irrelevant because the particle will always be crossing the maximum width of that laser, which also helps with the sensitivity we're talking about and just makes it more robust. So part of our collaboration that we've done with our friends at Thermo Fisher and the Invitrogen headquarters in Eugene, Oregon, have been assisting them to evaluate new bandpass filters. And we've been able to come up with a different filter that is normally supplied with the original instrument in front of the side scatter detector, and this has greatly increased the greatly increased the sensitivity of the system. I think that we and others in this collaborative effort have come to an agreement that the new bandpass filter really does make a huge difference. And as I mentioned before on the previous slide, the ultrafiltering of the focusing fluid really does dramatically decrease the background noise that we see in running samples. So how do we validate that a flow cytometer can actually see exosomes? Quality control material at this point in time primarily exists of microparticle beads, just like any other beads that you would use to assess the performance of your flow cytometer. But in this case, they're much smaller. Presently, the industry standard is considered to be the Apogee Flow Systems beads called Apogee Mix. They consist of a range of 6 silica particles and 2 polystyrene particles, which actually have green fluorescence embedded into them. The distinction between silica and polystyrene is that silica has, the material itself, has a refractive index closest to what the refractive index of true vesicular particles are. So it's really considered to be the best, closest reference standard particle that we can use. We also use the Biocytex Megamix particles. Those are also polystyrene with some fluorescence embedded. And we actually like to mix those two. On this histogram, you can see that the top 2 peaks are the Apogee polystyrene beads. I'll first start off by saying we generally do not see those matching any size of any other particle. I'm not sure why. We basically just kind of ignore those. But when you move down to the Megamix polystyrene beads, you can see 3 populations of 500, 900, or 3,000 nanometers, which is almost off scale. And the red boxes indicate that those polystyrene beads actually line up pretty well with the similar size silica particles. So the 500 nanometer Megamix lines up with the 590 nanometer silica particle from the Apogee beads, similarly the 800 to 880. Sometimes we don't always resolve that 1300 nanometer silica particle. Sometimes we see it, sometimes we don't. We're not really sure why. But again, I don't really care because I'm interested on what's going on to the left of that. So from that point on to 500, and we resolve the next smallest, which is at 300, and then we go to 240. We've got the 180. So around the 10 to the 4th hash mark is where we're starting to see those smallest particles, and that's going to be the range that we would expect to see exosomes appear when we run them on the cytometer. Much below 10 to the 4th is really pretty much considered electronic noise. The large orange portion in the corner is probably the electronic noise, and then perhaps that next cloud over would be our 30 nanometer background particles after doing the ultrafiltration. So as long as the beads look good, I say, yes, I think my cytometer can see small particles. Well, let's test that. So we're going to make some exosome fractions and do our successive ultracentrifugation spins. And far left, you can see where we've run just 500 microliters of ultrafiltered PBS. Again, small amount of electronic noise. That's run at a threshold setting of 200. And generally, we move that threshold up to about 500, which is where that red line is indicating, so that we can not count noise particles. And then the rest would just be the 30 nanometer or less debris zone in that PBS. When we run exosomes on the next panel over, we see that they are pretty much in that very small zone of what we would hope to be 30 to 150 nanometers. And for just points of reference, there's very little in that gate on the right. The next larger spin, microvesicles obtained from 16,500, show the concentration in this density plot moving to the right, so we're getting bigger, up to about 33% in that right gate. Then our apoptotic bodies, which were the first spin at only 2,000 G, shift that density plot even further to the right, and now we're at about 54%. So not only do we have confidence that we're seeing the exosomes, but the other things that we know to be in an appropriate size range are all falling into pretty much the correct size zone. So when we see things like this, we have fairly good confidence that the instrument is seeing individual exosomes, and they're putting them on scale pretty much in the right spot in correlation to our reference beads. Next, we have to make sure that the instrument is actually analyzing individual exosomes. This is critical. Many of our preps get highly diluted on the range of anywhere from 1 to 100, sometimes up to 1 to 10,000, depending on how concentrated these preparations are made. We generally take the pellets from the ultracentrifuge spin and resuspend it in a fairly small volume, which means that we then have to do pretty large dilutions to get to the point where we want to do this kind of flow cytometry work. To validate that we aren't seeing what we call a swarm effect, which would be multiple particles coming through the interrogation point of the laser at the same time, by doing this simple dilution series. So we make a starting 1 to 100 dilution, run it, and see that we get 5711 counts per second. Successive 1 to 2 dilutions of that with ultrafiltered PBS, in theory, should cut that in half. So the number in the center is the number that we actually acquire, and then the theoretical number on the far right corresponds to half that number, half of 5711, and then you just kind of cut those in half, and you'll see that those match fairly well. I'm sure that there are probably some acceptance criteria that of what percent variance can you accept as tolerable. To me, something like this looks perfectly fine. That gives me perfect confidence that we literally are putting only one particle past the laser at a time. And that's been one of the biggest problems in cytometry. So now we're able to actually resolve these individual particles. We got to make sure that we have our sample diluted enough that they literally are going by one at a time. This is a critical quality control step that we do with every single exosome prep that we make. So now I guess we need to figure out what we're going to do with these exosomes. What do we want to look for? We've got one more little quality control step to go through. So when you do this ultracentrifugation step, you do get some other things pelleting besides exosomes. So these might be small protein aggregates, but we've got to be able to exclude what would be true debris in that final spin versus true lipid bilayer vesicular particles. We do that using a lipophilic dye, which is going to allow us to really say that's a vesicle, that's not, and make that decision. So we have tried a lot. We've settled on a product from Thermo Fisher known as FM143 and FM464. These are lipophilic sterile dyes that have the ability to intercalate into the outer leaf of the membrane. My favorite part of this molecule is that it's not fluorescent until they actually intercalate into that outer membrane, similar to like a DNA dye binding into a DNA strand. So what this especially has allowed us to do is to develop a no-wash staining protocol. So we can add enough of this dye to stain, but it's not enough to cause problems with anything else. The debris particles will not pick it up. And so this has become our favorite dye to use for this lipophilic ID. The key point of trying to develop a no-wash staining protocol is that every wash takes about at least 2.5 hours. By the time the ultracentrifuge comes up to speed, you spin for 2 hours and you spin it down. There's no such thing as a quick wash when you deal with exosomes. So we've worked very hard on trying to develop our no-wash staining protocol. There's other lipophilic dyes you can use. Bodipi TR ceramide is actually just a lipid molecule with a fluorescent tag bound onto it that will also intercalate into the outer membrane of the vesicle. One thing I think I just realized I forgot to tell you is that the FM1-43 is excited by a 488 nanometer laser, and it emits its light in the 525 to about 625 nanometer range, fits into the PE area. And then FM4-64 needs green, the 561 nanometer laser excitation, and it's going to emit further down in the red range, 700 to about 700 nanometers. So you have two different color options there to choose as you also look for what antibody markers you want to put. We always do this in combination with antibody staining. The Bodipi is actually only available in one color. It uses 561 excitation and emits light in range of 575 to about 625. We have not really tried this with or without washing. It may be possible to use that one as well without washing, but we have not validated that. The other options, the DI series, which are actually carbocyanine membrane dyes, have been widely used for many, many years as membrane labelers to do in vivo cell tracking, cell mixing, cell fusion experiments that have been used for many years, as well as PKH dyes, which are similarly able to intercalate into the outer membrane. And PKH has actually been made into a whole rainbow of colors. I think there's actually 8 different fluorescent molecules across the whole span of the range that a cytometer can detect. The big problem with these two kinds of dyes is that you require multiple washes to get rid of all of the excess dye. They're really kind of a pain to deal with. It's what we started with. We started with the PKH, and one of my students was reading through a paper and saw the reference to the FM1-43, and we ordered it and tried it, and it was like, okay, never again. So that's where we stand. We believe that's probably your best choice, but there are many options there. Once you're going to do that staining, now we can say, what's debris, what's a true lipophilic potential exosome? Now we're going to use antibodies to look at 3 gold standard reference molecules, those being CD9, CD63, and CD81. You cannot call a particle an exosome unless it has at least one of those markers on the surface. Let me repeat that. If you don't see at least one of those 3 CD markers on the surface of the exosome, you cannot call it an exosome. So these are fluorescently directly conjugated antibodies. And we'll talk a little bit more about the way we do this. So as I said, we've got a no-wash protocol. So we are able to actually, even though we do DLS to measure the size of our exosomes, another benefit of the Attune NXT cytometer is that it uses volumetric counting. So it can give us a fairly accurate concentration of the particles in there. So when we're doing those dilution tests, we're also kind of validating what the actual particle concentration per milliliter is in that particular sample. So knowing that number, we aim to put about 250,000 exosomes in 250 microliters of ultrafiltered PBS. we generally just use standard 12 by 75 polystyrene wax tubes. We add our FM143 dye at a final concentration of 0.5 micromolar. And then we add our antibodies according to the titrated optimum concentration, which is generally somewhere between the range of 50 to 200 nanograms per mil. We incubate that sample at room temperature for 10 minutes and then immediately place them on ice and try to run them on the cytometer as fast as possible. It's a fairly straightforward staining protocol, and we've done a lot of work to optimize that, trust me. Titration of your antibodies is very important, as it is in all aspects of flow cytometry. One must really titrate your antibodies to do good work. So this is an example of what the lipophilic dye looks like. Center panel is unstained exosomes. I've popped up the reference of the beads just to kind of show you that we are in that range, around that 10 to the 4th hash mark. The threshold was set a little higher when these samples were collected, but we are right there in that size zone for exosomes. Then this gate on the right is going to actually show you the particles, a good percentage. I think we're 84% on that one. We do see some variability, so you could go anywhere from 50% on the low end up to maybe 90% on the high end using differential centrifugation of the purity, and that's why we do all of these QC steps for every single preparation we make. Differences in, of course, what the sample starts from, be it culture supernatant versus a body fluid, but even just you would consider fairly uniform culture supernatants can provide different purities in preparation. So it's really important to do all of these upfront QCs on every single preparation. We use this positive gate on the right to then look at antibodies. So we'll gate from here to the next panel, which is going to show us a 3-color stain. Now, we always use isotype controls, matched isotype controls, to make sure that these exosomes aren't too sticky. As I think I mentioned a few times earlier, the precipitation kits would provide such sticky exosomes that the isotypes were as bright, if not brighter, than the marker of interest antibodies that we were using. And so when we first started seeing that, we realized, okay, we can't use precipitation kits because the staining is meaningless. So by using isotype controls, we can set these quad stats, then look at the expression of our 3 reference gold markers. Top right panel shows that almost 90% of the exosomes in this preparation that were also positive for FM1-43, so this is gated from that positive lipid gate, are expressing 63 and 81. The bottom panel, when you do 81 versus 9, the 9 isn't as strong. Probably about 60% have crossed that threshold. Again, every prep can show high variability in how much of each of those markers are there. Sometimes you don't have any on one of them. You might only have, say, 9 and 81. And we do all 3 because we need to assess what that particular preparation has on its surface. So we'll pick one that has the preponderance. In this case, we chose CD63. And then we'll do extra staining tubes with 2 more markers of interest. So we'll put in our 63 antibody. And in this case, we're staining exosomes that we've isolated from the culture supernatant of cancer cells, Mia-Paca cell line actually, pancreatic cancer cells. And in this case, we're comparing cells that were grown in 2 different conditions, hypoxic and normoxic conditions. So we've done all of the spinning with all of our culture supernatant from these two comparative cell lines, and we're looking for the markers CD44 and CD184. And we actually saw quite a distinct difference in normoxic conditions. Not all, probably only, let's see what the number is here, 40% of the exosomes are actually staining positive for CD184. They're fairly uniform for 44. All of them have 44 on them. But in normoxic conditions, we only see about 40% 184 positive. And in the hypoxic conditions, and this is related to cancer stem cellness, the stemness of these markers increase in hypoxic conditions. And so now all of the exosomes are dual positive for 44 and 184. So those are just representative figures of the kind of data that we're generating. One quick comment on the choice of fluorochromes. You must strive to use only the very brightest fluorochromes, those being the polymer violet dyes, phycoerythrin, the Alexafluor dyes, especially 647. It's hard to find, but sometimes one of my favorites is also Alexafluor 546. It's also one of the brightest ones. But you really need good, strong, bright dyes because exosomes are small. They're probably less than 1/100th of the size, or an antibody is probably only less than 1/100th of a size of an exosome. So you can't really stick too many on the surface. So you need each fluorescent molecule to give out as much light as possible to be able to generate a signal that you can detect. So I'd like to wrap up today by letting you know that, in my opinion, exosome flow cytometry requires vigorous validation at several steps. You have to start off by characterizing your instrument and making sure that your background noise does not interfere with being able to resolve the small silica particles that are, at this point in time, the best QC material we have. I know several companies are working hard on generating what's going to be the next generation of QC material, which is going to be a stabilized small vesicle in the correct size, which is going to give us the ultimate material as a quality control standard because it's going to have the same refractive index as our exosomes. But for now, the silica microparticles are really the best way we have of assessing our instrument. So I've shown you that there are a variety of methods to isolate exosomes. I strongly encourage you to try several of them. It depends on what kind of starting material you're using. Plasma is much more dirty than, say, urine if you're going to go after a biofluid. Culture supernatant is pretty easy, and ultracentrifugation is pretty standard for that. But try lots of things. See what in your hands gives you the best result. This is not something, and I have to say this with the strongest emphasis. If you're thinking of starting working with exosomes, don't think you're just going to try it once and get some pretty data. We've been working on this for 2 years. This is not a trivial undertaking. You have to be very rigorous and methodical in the way you do things to really be able to prove to yourself and convince yourself that you are truly seeing the correct particle. So it's been a lot of fun, and I think that there's no question that exosomes are the wave of the future for potential diagnostics, for figuring out intracellular communication and ways to interrupt that might potentially develop new diagnostic or therapeutic effects for a myriad of diseases. We are very excited about the potential of this. We work predominantly with pancreatic cancer patient samples here, and that's the disease that we're going after. I guess I'll wrap up by saying our collaboration with the flow cytometry team at Thermo Fisher has really been wonderful. I think that our work together has greatly improved the confidence that this next generation Attune NXT cytometer has the ability to see microparticles and can reliably provide good surface antibody staining data. And I think probably the last thing I'll say is that the new side scatter filter that we've come up with will be available in the near future. It takes a while at a big company to come up with a new product and part number and everything, but it's coming, and you will have access to that in the near future. Just one small simple filter to buy to swap out on your instrument. And I think that's all I have. I thank you very much for your attention. I hope it's been beneficial, and I'd be happy to entertain any questions.
Thank you, Steve, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Steve McClellan will answer as many questions as time permits. First question is, what are the challenges of using polystyrene beads and silica beads as size standards?
So, you know, I hate to patently endorse one product over another, but Apogee really is the only company offering these small silica particles. There is a vast difference between the refractive index of silica versus polystyrene. It is generally accepted in the field that polystyrene does not offer a close enough true sizing standard to judge the size of an exosome. You must use the silica particles at this point. They really are the only thing available that's close enough to the refractive index to really gain a true sense of what size of particle you're resolving in your flow cytometer. And coupling that with the ultrafiltered sheath fluid really does make a huge difference in making that resolution happen.
Thank you. Next one is, did you consider instrument resolution limitations when designing your experiment, and what considerations should be accounted for and considered?
I think really you have to start at the beginning. You have to start with the beads and prove to yourself that your instrument is capable of resolving those beads. I'm quite sure that some of the stuff I've told you today can be applicable to other cytometers as well. But until you do it yourself and prove that you're actually resolving all of those little silica particles, there's no sense in even doing any exosome preparations. So it's you can't put the cart before the horse. You have to validate that your instrument can actually resolve things in that 30 to 150 nanometer range, and the only way to do that is by using.
Great, thank you. Next one is, what do you see as future directions in the exosome field, and what role can flow cytometry play?
Wow, so last week I attended the American Society of Exosome and Microvesicle National Meeting in Baltimore, and I was just astounded. It's been several years since I attended that conference, and I was astounded at how many different disease states are actually involved in exosome work. I mean, everything from Alzheimer's, neurodegenerative diseases, of course cancer, diabetes, restless leg syndrome. I was astounded to see their poster on restless leg syndrome, and they think that they can now predict which Parkinson and Huntington patients will be affected more heavily by that, just looking at an exosome signature. It was just astounding. I have no doubt, like I said earlier, that this is the wave of the future. There are so many potential uses for these things as early diagnostic markers, predictors for potential therapy solutions. The sky's the limit, and as I said at the very beginning, if you look at in PubMed, it literally has been an exponential growth of reports of people looking at exosomes. Cytometry is going to have a much bigger field, part to play in this field, especially if we can move towards generating cell sorters that are more sensitive. It has been done. That's even harder. Oh my gosh, I can't even imagine. But if we can then push to that point, so we can now do this surface labeling and then sort out a specific marker on the surface and see what's inside of maybe those exosomes. Sky's the limit. It's a very exciting field. Excited to be in part of it.
Thank you. Next one is, how were your samples collected, and were there any time restrictions?
So, for working with culture supernatant, it's not that difficult. You do have to start with much larger volumes to get the number of exosomes back. So on average, we would do all 6 places in the ultracentrifuge rotor with 50 milliliter tubes. So we would try to spin at least 300 milliliters of culture supernatant to start with. That generates, in general, a pretty good amount of exosomes to work with. The interesting thing we've noticed along the way is that there's huge variability in the number of exosomes that different cell lines secrete. Some don't secrete very much at all, and others just make hordes of them. So that's something to consider as well. You might want to try several different cell lines to see what gives you the best yield to have material to work with. I think I mentioned in the beginning, we use patient blood, and that's collected in a green top heparin tube as plasma. It is critical that you do not use serum because the process of letting blood clot traps so many of the little exosomes in the actual hard clot so that when you just dump out the serum, there's very little exosome left in it. So you really have to use plasma. Many of my colleagues use urine, especially looking at prostate cancer. That's a very nice way to isolate exosomes because the urine's actually pretty clean and pure of other things. And again, you can use cerebrospinal fluid. There's lots of different things you can do. One thing that we did test early on and that we feel is very critical for obtaining blood from patients is that we want to stabilize that plasma with a protease inhibitor as fast as possible. We strive to have that tube of blood in our hands within 30 minutes of it coming out of a patient, spin it down, and we immediately add a standard protease inhibitor tablet to a certain concentration, whatever is recommended. I'm not even sure where we get ours from, but that's a pretty standard thing. But we've done this side by side. We've done time tests. The blood sat at 30 minutes to an hour to 4 hours with or without protease inhibitors. And we do see a strong loss of surface markers on the exosomes if we don't have those protease inhibitors in there. I probably should have mentioned that in the main part of the webinar. It's very important to have those in there to help preserve the structure of the exosomes.
Great. Thank you for that. The next one is, I'm really confused by terminology. EVs versus MVs versus exosome versus apoptotic bodies, etc. What is the standard terminology for small particles?
It's kind of crazy. I really think it's best to maybe go back and look at that slide where I've got the 3 successive spin speeds lined out, and because that's the best way to just wrap your head around it. So at each higher level of G-force, you're going to pull down a smaller and smaller particle. So the largest of these are considered apoptotic bodies. These are blebs that are spit out of cells as they're undergoing the final processes of apoptosis, where the cell is literally ready to break down. I mentioned large oncosomes, which are somewhat controversial in the field, actually, but those are kind of lumped into that large category. Medium category is always going to be considered a catch-all. Microvesicles can be anywhere from about 150 up to 1,000 nanometers. That's a pretty big size range. We do believe that the way they're generated is pretty uniform from the budding of the exterior of the membrane. And then an exosome is never bigger than 150. Some people are a little more strict and call that 100 nanometer on the top end, and say 30 to 100 nanometers. So we're pretty confident in how those are developed as well, with the invagination and then the multivesicular endosome body that then refuses to the membrane and releases those little particles out to the surface. There have been groups that have done just phenomenal live cell imaging. You can actually pre-label the membrane with some of these dyes. I talked about the lipophilic dyes, and then literally over time-lapse videography, watch exosomes being released from cells in live time. It's a phenomenal thing. So we know how it works. We can watch it. And really, you just think about small, medium, and large. That's the best way to think of it.
Well, thank you. Looks like we have time for one more question. How did you determine the optimal titer of antibodies to use in this application?
Really no different than titration of antibodies on cells. It's very important to try a variety of different concentrations. What you're looking for is a signal-to-noise ratio where if you have antibodies at too high of a concentration, the negative cells will kind of creep up and be brighter. Optimally, what you're trying to do is, and the process of titration really is to maximize the space between the negative and the positive population. And so when you do your titration of different concentrations, that's what you look for. And then at a certain point, of course, you're going to start to drop off your signal. So you know that's gone too far dilute. There are many, many references to talk about how to properly do titration of antibody out there and just search the web, and I'm sure you can find 4 or 5. But it's very important to try titration with all of these epitopes, especially in our case where we were trying to minimize the amount of antibody to get a signal in this no-wash particle. So we found that to be very, very critical. Can't just go adding a whole bunch of antibody in there and not wash it back out.
Well, thank you. I would like to once again thank Steve McClellan for his presentation, and I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through May 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.